Indonesia One Health University Network, or INDROHEALTH. This is something new, and I will be here from him what is this new institution in the university of Bogor University. This topic will be reflection on one concept for animal and human diseases or zoonosis. Ladies and gentlemen, we will have each of these speakers for 20-minute speech, and I would like for keep in line so we can have some time for discussion in the last session of this one. So let's start with Lisa. Go ahead. Thank you.
Yes. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Thank you, Mr. Chairman, Professor Didi Hajar Gunadi, for introducing me. So maybe I would like to take some of your time, 20 minutes, because Taco gave me 20 minutes. So I will try to be brief and concise about what I'm going to present. I will introduce myself after this slide, but first I would like to let you know about the potential application of the no-self DNA technology for agriculture in Indonesia. But for disclaimer first, because in Indonesia this is still also a new thing, Professor Mazzoleni. We learned so much from your research and your team that you have done in Naples. So we would like to show you all a brief summary of what we have done in Bogor. Actually, we are a team of work. Myself and Galuh Permatasari and also Diniastika Sari, Imam Bagus Nugroho, which he was no longer with us because he moved to other institution, Professor Didi Hajar Gunadi and Mr. Taco. Okay, allow me to introduce myself for a little bit because basically I'm a scientist, so what I am going to present you is all about based on data that I have seen in my laboratory. So I have my expertise mostly on molecular biology, genomics, of course, working on a lot of DNA sequence in plants, in humans also. When I was in Beijing Institute of Genomics, we learned so much about how to deal with the human genomes that is really different with the genome from plants or even microbes. And during my PhD, I learned so much and studied a lot about the genome of rubber tree, so Hevea brasiliensis. So basically, I'm a laboratory guy looking for a sequence in front of my computer, a big screen normally, and try to understand the meaning behind every sequence of genome or even in a genetic aspect. So for the last 2 years, I worked a lot with many partners, and thankfully, Taco invited me. Thanks also to AND Biopharma for giving us the opportunity. Professor Mazzoleni also giving us the opportunity to learn and to study about the no-self DNA technology. Okay, so a quick reminder about what is a self-DNA mechanism. So at first, when Taco came to us and talked about this mechanism, because I was with Dini, Taco, you remind, I think you you still maybe remind of our faces, right, when you talk about this mechanism, because I'm a molecular biologist, learning so much about DNA and the mechanism. Even we try to understand what is about the degradome, how to understand the sequence that already been destructed by the microRNA in plants. So when you talk with us about double-strand DNA that fragmented and give the what we call the effect to the plants or the organism that absorbed it, it's bubbling in my mind, because as a scientist we need to talk about the theory that should be proven by a data. So in this mechanism that Taco once talked to me and give me the paper, he tried to show me this one, that a self-DNA concept is that a inhibitory mechanism of DNA happen to the conspecific species. What is this conspecific species? Conspecific species is that the DNA came from its own organism. For example, if you want to inhibit yourself being lazy, for example, you should take your own DNA and ingest it. This is only an analogy that maybe can make you understand better the concept of the self-DNA. Basically, when you have grass on your field, on your houses, and you mow the grass you will smell something like a weird planty smell. That's what we call allelopathy concept. Actually, this is a volatile organic compounds that went out from the plants, goes into the grass maybe 200 or 300 meters after, and the grass over there can understand, this is some type of a communication between the plants. That grass will understand that this part of area has something wrong. For example, if the grass has been attacked by a pathogen. So they told the grass, the next door, to enhance yourself, maybe do some fortification on your cell wall, or just enhance your self-development so that you cannot waste the energy or you can absorb or do something like developing a rhizome so that you can protect yourself. That kind of communication happens and already known for many years, what we call allelopathy. But self-DNA concept is perhaps for some people quite difficult to accept because you need to prove it, right? You need to prove it. How they say that the DNA specificity is quite specific between the same range of species. Longevity means that you can go with long time. But as Professor Mazeroni mentioned in the earlier discussion, that we need to understand also, we don't know yet how long the DNA of self-DNA can be accepted by the plants or accepted by the organism. And another quick reminder is that a part of self-DNA, that DNA that inhibit the growth of its own organism, we have also the concept of no self-DNA, meaning that when you go around to the soil, you have a decaying plant, and this decaying plant releasing its own self-DNA, but you have also other organisms around that also decayed, and then they release also their DNA to the soil of the environment. And that DNA, the red one, the one that is not originated from the decaying plant, will also have an influence to the plants nearby. So that is what we would like to understand between, when you talk about organism, you talk about homeostasis, it's the balance between the good and the evil, the balance between the self-DNA or no self-DNA. But I did not say that the self-DNA is bad, no. Because what we know right now is that the mechanism is inhibitory. But maybe we can find, maybe in the next future, another mechanism that perhaps even more complicated than this one. I think Professor Mazeroni already thought about this also I'm quite sure. So we need to understand that in the world of science, what we know now may be changed in few hours or even days or months or years later. But the concept of this self and non-self DNA, meaning that if you try to understand the no-self DNA concept, is an inhibitory mechanism of a DNA that comes from the non-conspecific species into the targeted path. I will explain to you in the next slide. I think I forgot who asked Dr. Romney, right? You asked about how the nucleic acid can be sent by the organism. So you talk, this is from the Nature Review by Slee and Hartman in 2016, talking about not only RNA sensing, this one, double-strand RNA or single-strand RNA, but also double-strand DNA sensing, meaning that all mechanisms are possible. What you need to understand is that this is the case in the human or even in animal. When you have double-strand DNA, depending on the receptors, even though even the receptors normally related to the immunity system of the organism, when you have this double-strand DNA, it can trigger many mechanisms. For example, immune sensing receptors or even nucleic acid receptors. And then you have a lot of aspect that is going on the organism. When you talk about this in animal, I think the same in the plant. Maybe the mechanism is not quite maybe 100% alike, but the concept should be the same because you have receptors in every organism and then you have activities. So that is why in our laboratory, we try to understand this concept. So this is thanks to, for that moment, we have a research group what we call ALIBI, aligning bioinformatics and also working closely with PT Ecotalis Technology Indonesia and also Biopharma BV and corporate system. We try to understand the mechanism. We try to duplicate or try to reprove what Professor Mazzolini's and team has been done in Naples. So what we basically do, we work with two species. The first one is Oryza sativa, rice. Second one is the oil palm. The next slide, I will explain to you about the oil palm. But first, with this rice seed, we try to treat them with three applications. First one, we only applied with this cotton under a small petri dish using a distilled water. It's a sterile water. And then we try to wait for the growth for 5 days. But actually, we continue the counting until several weeks until the plant died. But in 5 days, you already can see that there is some changes. The second one, we use no-self DNA using a Xanthomonas DNA. Xanthomonas is the pathogen that attack rice. So it came from different aspects of microorganism. And then the last one is self-DNA from its own, a rice leaf. What you can observe is that there is a normal growth in the control. No problem, everything looks well. But when you see no self-DNA using Xanthomonas DNA, there is something changed. You see, this one, the green one, is what we call plumula. Plumula is the leaf. And the white one is radicle. Radicle is the root. What you can see here is that you have particularly smaller numbers of plumula, but very long radicals. But when you apply itself DNA here, you see there is a very low and slow growth of the even radical or also the plumula on its own self-DNA, meaning that the theory, the previous 3 slides, has been proven here. So we believe that something happened here in no self-DNA using Xanthomonas that triggers something in the rice seed. So the rice seed order the cells to growth and to maybe try to seek nutrient for defending themselves. For example, this is a theory, That's why the roots grow very long. And the next aspect is we learn in the Elais guinensis in oil palm. We use a seed from the oil palm. We experimented the same thing using no self DNA, this one, no self DNA, using its own famous pathogen, Ganoderma boninense, and using also the environmental DNA. What we call environmental DNA is that it's a combination of a lot of DNAs in the soil. So we name it a soil DNA, and then we have this normal growth, only distilled water, and then this one self-DNA using leaf from the oil palm. So we use both leaf, so that the reason is that we need to compare between rice and also between oil palm. What you can observe is that you observe the same tendency or mechanism. When you have no self-DNA, you have enhanced root growth. When you have environmental DNA, you also have enhanced root growth compared to the normal growth in the distilled water. But when you apply self-DNA using leaf from its own species, you have approximately undergrowth, differences in few centimeters of a long and numbers of lateral roots. So meaning that the no self-DNA gave again a different phenotype compared to the self-DNA. So using these two small and in vitro experiments, we would like to prove that the no self-DNA mechanism could be beneficial to the organism that we apply to. And we would like to come back, make sure if really true that the self-DNA itself can inhibit its own organism. So we use both of the pathogens, Ganoderma, this one, Ganoderma, and also Xanthomonas. We use a small microplates with hundreds of tubes. So we take pictures only 6 of the tubes. It's very small tube. We use spectrophotometry technology to really make sure that the obscurity of the liquid is not based on the media, but based on the growth of the pathogens, of the microorganisms. So what we can see here, this one is controlled. When you see yellow, because there is a peptone in the media, so you see yellow, but actually it's sterile. And then we apply sDNA here by its own. This one, so sDNA from Xanthomonas. This one, sDNA from Ganoderma. And this one, we did not apply sDNA, we just put distilled water. Actually the same with the control, but the control is only in media. So what you can see here is that visually you can see changes that, especially in Ganoderma. This one is mycelium, like right above the liquid. When you have a liquid media, you have a fungi, it tends to grow in the upper level of the media. But when you see here in the sDNA of the Ganoderma, the growth was inhibited. The same happens to Xanthomonas, the growth inhibited. It can show by the curve that we observe in our laboratory that has a decrease of number of colony. But when you plate them in the petri dish, the bigger petri dish, you can see that when you apply sDNA, this one you cannot see, but you have full colony. Every inches of the petri dish full of Xanthomonas, but when you apply sDNA, they tend to grow really slow, meaning that the sDNA concept is inhibited the growth of a conspecific microorganism, meaning that its own DNA inhibit the growth of itself. And how can we direct the no self-DNA technique? So many scientists, I say many because there are groups of scientists, a part of Professor Mazzoleni is also trying to understand this concept, maybe a bit different understanding, but the way they treat the experiment looks alike. For example, Feriskia Jimenez from Mexico, tried to put Phytophthora capsicum from pepper. From pepper, tried to extract the DNA and try to fragment it, even though they use different method from sonification, and they drenched the application of this Phytophthora capsicum to infect the pepper. And they saw a lower disease severity and induction of plant immunity, meaning that this theory should be improved and should be able in the future to be applied in the bigger level. And the question is that, because when you talk in vitro, you talk about what we call aseptic environment. You only several aspects that you can control. But when you go to the environment, not environment, just go to the greenhouse like this, you will see a lot of things like mixed organic sources, crop wastes, that you need to understand all of that. You can extract the DNA and you can call it environmental DNA because it comes from different species. And we don't know the effect of this environmental DNA. But Carvajal and Valenzuela tried to understand this and give the opportunity to create an experiment from it, try to induce the plant, what we call plant immunity. So the concept is basically like when we have a vaccine of a COVID-19, so that in the future we cannot get the disease to be severe. So the question is that us, as a scientist, we need to pay attention to the dose of the environmental DNA. I think that is the question of the day from fellow scientists. How much enough and how much is too much? That we need to understand importantly. And from the study of Professor Mazzolenis and team and other scientists, we understand also that feasibly extraction protocols need to be established. We understand that when you have application from DNA, you need to extract the DNA. How much you need to extract? If you want to apply, for example, in our institution, in Oil Palm Research Institute, you have hectares of plantation. If you want to apply that, how many DNA you will need, and how to administer that. That is the one that we need to think in the larger scale. The second one is that the utilization of this no-self DNA can be applied to handle plant pathogens. The third one is that how to see that is an extremely important concentration dependent. 200 ppm, 300 ppm for this species and 400 for that species. And that is the one that we need to think about. So Marcelo Llanos said to us earlier this morning the importance of factors, maybe microalgae, why not other microorganisms, why not other particles, for example, nanoparticles. If you have expertise on that, we can try to experiment on that and maybe we can see a good result. And massive duplication of DNA, maybe we need NGS machine, but then when you talk about business NGS machine, it becomes quite expensive because you need to produce something from expensive machines. Then when you goes on commercialization, should think about how the BEP will come back to us. And maybe to end the discussion, I would like to maybe try to give you a perspective of how we use this technology in the future. Maybe I will talk first in the blue line. So the blue line is quite clear. So the theory has been proven. We need to also enhance the experiment on a larger level, but we know that maybe we can use the no-self DNA application to try to overcome the problem of Ganoderma in oil palm or even Xanthomonas in rice that has been devastating for the last few years by developing no-self-based product. And the other one is how we think about the management of the oil palm plantation, for example, because we tend to use chipping and sunning for replanting. And I talked to several experts this morning, and they don't know how long we should put that biomass there. 2 weeks, 3 weeks, 5 months. Some of the plantations put there for 2 years or 3 years without doing anything. So we put back a lot of biomass, and of course we put back a lot of self-DNA in the soil. So I think that is all from me, and I would like to quote Albert Einstein, reminding myself as a scientist that creating a new theory is not like destroying an old barn, but actually it's like a mountain gaining a new and wider view. So I think that maybe no self-DNA is still new to us, but I think we should open our mind and, I don't know, the research is always something new. Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Risha Ariputranto, who has delivered the topic regarding the proof of concept about the application of no-self DNA to mitigate or combat the severe disease on both major crops in Indonesia, such as oil palm and rice. And he has shown also the potential use for Ganoderma in oil palm and Xanthomonas in rice. However, he also shows something that we got to take care about, because his finding will turn around the current practice, especially for the use of compost that originated from the same material of the biomass. This is something that's very challenging. Thank you, Dr. Isa. And on the second speaker of this afternoon, we do have Dr. Romi Roviadi, who is going to share that something different with Dr. Isa. It is related to fishery in Indonesia. You got 20 minutes, Dr. Romi. Thank you.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. First of all, I would like to say thank you to Ecotalis Indonesia for giving me this opportunity, and the organizing committee for giving me the golden place like this to present my recent findings on the self-DNA and the use of this concept in the to inhibit the growth of the Vibrio in aquaculture production system. All right, probably some of you do not know about aquaculture work, because we talking about plant and agriculture before. So I just want to draw you several attention about the aquaculture and fisheries work. We are the maritime axis right now, and we know that we have more than 17,000 islands and 108,000 kilometers square of coastline. We have more than 275 million people, and the majority live in the coastline. And we have 12.1 million tons of the fisheries production. And our ministry said, in order to build and establish the industry, aquaculture and fisheries, we need to make it balanced between the economic and also the environmental situation. So this is the goal for the fisheries and aquaculture production system in the future. So globally speaking about aquaculture, before we go into detail about the self-DNA concept, we know that in the future, 2030, World Bank already stated that 2/3 of the seafood product will coming from aquaculture. And even the economists say that aquaculture and not the internet represent the most promising investment opportunity in the 21st century. And look at the data from this FAO. We know that the aquaculture right now is already produce more seafood compared to the capture fisheries. So aquaculture is the future. So, and this self-DNA probably can be a part of this grow production system. And this is about the Indonesian aquaculture production. Here we have, in total, in summary, we have an increasing value, increasing number from 4.3 million tons. Right now in 2020 is of around 5.2 million tons. But there is a challenge on how to optimize the production system. We know that with the rise of the fuel right now, aqua feed, the feed is become the first challenge because I already talked with several feed millers. They will increase again the price of the feed in this October. So this is make the life of the farmers become difficult. So the aqua feed, the cost is become the first challenge, and the disease is the second challenge in order to optimize the aquaculture production system. Market price, because of this volatility and fluctuation in price, also become a challenge, and broodstock and another feed. So quality of the feed is also become the challenge in aquaculture production system. So among the infectious disease, there are a lot of disease, viral disease, parasite disease, bacterial disease. Vibriosis has become a cause of serious losses in aquaculture. This disease reported as a significant challenge to the expansion of the aquaculture and also cause economic losses, more than $100 million global wide, and high number of farm closed, and large number of job losses, and will take years to recover because of this disease, the Vibrio. So this is just to give you the idea about the number of Vibrio during the white phases syndrome in the shrimp. We can see that the number of Vibrio in the normal situation without the disease incidence, this is lower compared to the number of Vibrio during the white phases syndrome. So if we look at the intestine status in the shrimp that culture in the normal situation compared to the shrimp that culture in the white phases disease outbreak, we can see the high number of the Vibrio compared to the shrimp that culture in the normal environment. This is very interesting because Vibrio can be act as the primary causative agent or become the secondary causative agent. And current practice to manage the Vibrio, there are several practices right now that we can apply, the farmer can apply. So there is a prophylactic approach. We can use the immunostimulation, vaccination, but vaccination not for shrimp because shrimp is only rely on the non-specific immune system. This can be applied in fish, probiotic and quorum quenching. Biosecurity is also to minimize the disease incidence, feeding management, and also the water quality management. So there are a lot of water management system, the biofloc, semi-biofloc, autotroph, and all of these things is about how to manage the microorganisms in the culture environment. So we're going to self-DNA. We're already talking about the self-DNA, about the functional role of the self-DNA to also to inhibit the growth of the pathogen. So we have a thinking, that idea, to assess the species-specific inhibitory effects of the self-DNA from Vibrio harvey and also the Vibrio parahemolyticus, and also to test the species-specific inhibitory effects of the self-DNA to the growth of Vibrio inside the Artemia. You know, Artemia is the very important live food in aquaculture, and other than the microalgae, rotifer, Artemia is the most zooplankton that used in the hatchery right now in aquaculture production system. So this is the material and methods that we developed to prove the concept of the self-DNA in the aquaculture. So we have a pure isolate of the Vibrio harvey and Vibrio parahemolyticus thanks to the Gadjah Mada University that provide this isolate for us. And we extract the DNA. This is a normal extraction process. And after we extract the DNA, we electrophoresis the DNA, and then we come to the sonication. We cut the length of the DNA from 10,000 Besper to 100 Besper with these tools. We use the ultrasonicator machine, which function to fragment the DNA. So we fragment the DNA into small size from 10,000 Besper until 100 Besper. So this is the agarose gel looks like. So before, when we extract the DNA, when we extract the DNA, we have 10,000 Besper, the length of the DNA, and then when we fragment them with the sonication process, then we can cut the DNA into 100 Besper here. And for the in vitro trace, we developed this protocol. We use several dilution. So after we fragment the DNA, we also measure the amount of the DNA with the nanodrop spectrophotometer, and then we got the number of the DNA of the self-DNA is around 96.3 10 for the Vibrio parahemolyticus, and 53.33 nanogram per microliter for the Vibrio harvey. And then we make a stock solution. The stock solution, because we have a 100 microliter of the Zobel medium, and then we add another 100 microliter of this self-DNA, so the concentration become dilute into half. So the final concentration of the self-DNA is become 48.05 for the Vibrio parahemolyticus, and also 26.67 nanogram per microliter for Vibrio harvey. So we put this in the dilution factors, and then we use this well to see the inhibition effect of the self-DNA. So this is the in vitro test using the Vibrio parahemolyticus. You can see, as you can see in the slide, this is the control without the self-DNA. When we put the Vibrio harvey, the Vibrio parahemolyticus inside the Zobel medium, they grow fast because this Zobel medium is a specific medium for the Vibrio. But when we add the self-DNA here, we add, with the 48.0.5, there is an inhibition. So they suppress the growth of the Vibrio parahemolyticus inside the medium significantly lower compared to the control treatment. And then there is another concentration that because we have egg dilution factors, then the second dilution can also inhibit the growth of this Vibrio parahemolyticus. But at the end, this is something that I asked to Professor Stefano this morning. What is the minimum amount of the self-DNA that can work well to the inhibition factor? Because our trial to prove this concept, when we go down, lower concentration level of this self-DNA, so the inhibition factor is also going down. So the number of the pathogen is keep growing and growing. But with a certain amount of the self-DNA, we can see that there is an inhibition here. So we also prove this concept in the Vibrio harvey. So in the Vibrio harvey, we also have the control without the self-DNA, and then this 26.67 of the nanogram per microliter of the self-DNA, and we go for several dilution also. And here we can also see there is an inhibition here with the Vibrio harvey with this amount of the self-DNA. And then also there is even statistically, this is not significantly different, but in terms of the numeric value, there is a difference here between compared to the control treatment without the self-DNA. So it works. It works, the self-DNA, to inhibit the growth of the pathogen. And this is very interesting for us. And then we also think we do not want to stop until here in vitro test. We want to go further, because aquaculture is really big industry, and we need to take a part to this industry and then prove this concept into the industry. So we are thinking to use the Artemia. As I said earlier, Artemia is a very important live food in aquaculture during the hatchery, during the larva stage, and they are filter feeder. They consume everything, including the bacteria. So in this, so it is what it is. Whatever you feed the Artemia, that is come to your larvae. So when you feed the Artemia with the bacteria, then you also feed your larvae with the bacteria. So this is why we use the Artemia as the important live food, and also as the non-selective filter feeder. So before we go to the Artemia, this is just to show you the visual. There's the quantitative number. This is a visual with the Vibrio parahemolyticus and also Vibrio harvey. You see that if the color remain blue, meaning that there is an inhibition growth, but if the color change from blue into the pink color and colorless color, there is no inhibition. So we can see this actually it can prove the concept that there is an inhibition. This is to confirm the result of the quantitative number. Here they remain blue here for Vibrio parahemolyticus, meaning that there is inhibition factor there with the self-DNA. And also the Vibrio harvey, these two concentration level also have the inhibition effect to the growth of the Vibrio harvey. So this is what I'm talking about. So in order to get a very good design of research, we culture the Artemia in the axenic condition. Axenic condition meaning that there is no contamination. Only that type of bacteria that we want to investigate, that are present in the environment. So all equipment, including the Artemia cyst, including the culture water, including the equipment, we sterilize them. So there is no contamination there. And we use the axenic larvae Instar2 nuclei to prove the concept of self-DNA. And this is the axenic condition. After we harvest the Artemia, this is the Instar2 Artemia looks like, after we harvest them in the sterile condition. And then after we decapsulate the Artemia, we immerse this Artemia in the self-DNA, and then we challenge them with the Vibrio parahemolyticus with the initial concentration. So we have 48.05 and Vibrio harvae 26.67 nanogram per microliter. And then we challenge them with this pathogen and see what is the number of this pathogen inside the Artemia. So this is the challenge test. This is very interesting. So this was performed by my master student in Gadjah Mada University, and they see that the survival rate of the Artemia immersed in self-DNA, they have a better survival rate compared to the Artemia that do not immerse in the self-DNA. So also in the Vibrio parahemolyticus also they have a good survival rate, better survival rate compared to the control treatment. And about the density of bacteria during the challenge test. So we also observe, we collect the Artemia in the several time points. We see what is the density of the bacteria during the challenge test. We can see here that the density of bacteria in the Artemia without self-DNA is higher compared to the Artemia that immerse in the self-DNA. So this is the density of bacteria during the challenge test. This is with the Vibrio harvae, and this is for Vibrio parahemolyticus. So from 12 hour until 48 hour, the density of the bacteria is always lower in the Artemia that immerse in the self-DNA compared to the Artemia that do not immerse in the self-DNA. So the thing is, we not prove the concept, but we already apply the concept. So we can see the concept is work in this aquaculture production system. And this is, I think, is become the new approach how to mitigate this disease in aquaculture. Because, you know, aquaculture is really unique because they stay in the water. There are a lot of pathogen there. So why we not save the farmer by, you know, increase the survival rate and also apply this concept in aquaculture production system. Right now, farmers, especially for shrimp industry, if you ever heard about the disease outbreaks, there are a lot of disease in shrimp industry, and farmers waiting for the solution. Antibiotics is not the solution. So, but we have to move further in the safest way, how to save the farmer, how to increase the survival rate, and how to make a better, good return on investment in shrimp industry by using this concept. So, future study. Because this is also interesting. If this is work in the invertebrates, because Artemia is invertebrates. Invertebrates means they do not have the non-specific immunity, meaning that they cannot produce the memory. They are very primitive animal. But this concept can be worked in invertebrates. How about fish? Fish is vertebrate. They have a specific immune system. They can produce the memory. Probably the result will be different or probably better. So this is very interesting because our aquaculture, as I mentioned earlier, aquaculture in the future will produce more seafood. We are rely on aquaculture to get, because this lunch we already get a fish, a lot of fish, and in the future it's always coming from aquaculture. It's not from capture fisheries. But this should be started with a good milestone, a good step that we provide a solution to the disease, because disease can cause highest number of mortality in aquaculture. So I just want to highlight about the Vibrio because they are, the opportunistic bacteria. We also analyze about the communication thing about the bacteria. This is what they call as the quorum sensing, and there are several active substances that can also inhibit the communication of the bacteria of the Vibrio, what we call as the quorum quenching. So probably this is more advanced because we not only inhibit the communication, but we also inhibit the growth of the Vibrio in the aquaculture production system. So I think that's all for my presentation today. I just want to thank to Gadjah Mada University as our collaborators and Ecotalis Indonesia for collaboration. Thank you, and thank you.
Thank you, Dr. Romi Nopriadi. who has shared to us the current update of finding on the no-self DNA potential application on the fisheries, especially for shrimp with related to the febrile cause disease. Our last speaker this afternoon session is this kind of a reflection of what we have heard from this morning until this afternoon. Will be presented by Dr. Joko Pamungkas with the title of Reflection on the One Health Concept for Animal and Human Diseases. Dr. Joko, the time, 20 minutes.
Thank you, Professor Didik. Good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Salam.
The title given to me is about reflection of the One Health in considering the application or implementation of the self, no self technology in the One Health concept. And probably, as the name implies, I will be the last speaker, Pamungkas. And I probably have to start with the outline, but I will go very briefly with the definition of the One Health. I don't want to give an impression that I teach a basic knowledge of One Health. I believe that you all are aware about this concept or approach. And then also in relation with the zoonotic diseases, wildlife is very important in the One Health systems. So basically, we also would like to point out about the importance of wildlife. Also that in some aspect of the health, both for human health, animal health, and ecosystem health, we also see the benefits of applying or implementing One Health approach. And then I probably will give some success story of the global examples of One Health approaches that's been implemented also in Indonesia. And last but not the least, this is the very important things. I'm also new in this approach or technology of self, no self mechanisms. So basically, we also try to formulate some questions for the future application or implementation of self, no self technology in One Health concept. Yeah. Okay. For the definition of the One Health, we try to have the newest version of this definition. Actually, it's been developed by tripartite in the past, consisting of the WHO, FAO, and also the OIE. Now is called the World Organization for Animal Health. So the tripartite actually now joined by another organization, the UNEP, UN Environment Program. So then they actually have the quadripartite programs. As a visual definition of the One Health, basically on the left circles, there are the key sectors and also disciplines that are taking part in the One Health concept, such as the human health or human disciplines, and then also the animals as well as the environment. So the three sectors are actually taking the most critical part, but we are not ignoring, we are not eliminating the other ministries or the other sectors part other than these three key sectors. And then also, we also need to emphasize that this concept is not going to have benefit if it is only in the national concept or in the national level, but we have to also apply or implement in the subnational and particularly in a very, very spot or location that might be impacted by the interactions of the human, animal, and environment. Yeah. And then the circles in the middle is the outcomes of this approach, that we would like to achieve the healthy humans, healthy animals, also healthy ecosystems. And by having all these outcomes, we need to also consider the 4 C's: yeah, collaborations, communication, coordination, as well as capacity building. So basically, we just want to give you ideas that nowadays we cannot just work alone. Yeah. So medical doctor will not be able to actually solve human or public health issues without the help from the other disciplines or professions. Okay, so Indohun, not Indohan, probably. So basically, Indohun is an abbreviation from Indonesia One Health University Network. And it is part of the Southeast Asia One Health University Network based in Bangkok. Other than Indonesia, there are 8 now OHUNs in Malaysia, Thailand, Philippines, Myanmar, Vietnam, Cambodia, Laos.
Recording in progress.
We have about 20 universities in Indonesia as members of the Indohun. And out of these 20 universities, we have about 30-something faculties or schools. And out of the 20 universities, some already form or establish the One Health collaborating centers, like the University Gadjah Mada University also has the One Health collaborating centers, and then secondly, Udayana University in Denpasar, and third, the Erlangga University in Surabaya. The fourth is the Shah Kuala University in Banda Aceh, and the last one is the Cendrawasih University from Papua. And probably this year we will have the 6 OHCC, One Health Collaborating Centers, from IPB University, and next year probably from the Hasanuddin University, South Makassar, South Sulawesi. Yeah. Okay, so One Health is not just about infectious disease. Again, it is much broader than infectious disease. It's also inclusive the communicable disease, non-communicable disease. But I just want to point out here that talking about zoonotic diseases, so we have to consider about several facts that there are some infectious diseases that only affect human beings, and there are some only affects the animal beings. But also we have some zoonotic diseases that will affect human as well as animals. It can be transmitted from animal to humans as defined by anthropozoonosis, but it can also be transmitted from human to animals defined by zooanthroponosis. So in this case, we need to understand, we need to learn more about who is playing as natural host or reservoir and who is playing as fatal host. In many cases, the human beings are the fatal hosts from the reservoir role by the animals and also especially from wildlife. This again, another terms that probably is also heard very often, emerging infectious disease. So 75% of the emerging infectious disease in human are originated from animals. So this is the one that we call zoonotic diseases. And then out of these zoonotic diseases, about more than 60% associated with wildlife. This makes important that the approach must be a One Health concept, One Health approach. Yeah. This is a definition by the National Institute of Allergy and Infectious Disease, US NIH. Infectious diseases that have newly appeared in population defined as an newly emerging infectious disease, and then the infectious disease that have existed but are rapidly increasing in incidence or geographic range within certain period of time. this would be the re-emerging infectious diseases. Now, these are some points that makes us realize why wildlife are important in the One Health concept or One Health approaches. So again, more than 70% of emerging zoonotic infectious disease have wildlife origin. So if we try to give some examples, in 1981, we understand that the first case of AIDS are identified in human beings in Los Angeles. Yeah. And then 2 years after that, 1983, the virus was identified, HIV. Yeah. And then later on, maybe about 3 or 4 years later, SIV was isolated. SIV is the simian version of the HIV, of the retrovirus. Simian immunodeficiency virus was isolated later from non-human primates. But when we try to actually look further in the bioinformatics systems, the scientists actually proved that the SIV existed earlier than HIV. And SIV originated from non-human primates, especially from African species like chimps and also the talapoin monkeys. Yeah. And more pathogens actually are spilled from the wildlife to livestock, from the wildlife to human beings, or maybe from livestock to human beings. And this is because of the human activities creating more opportunity for the changes of types of contact that probably the several industry, mining industry that has to open lands, and also some development that has to actually open the forest. So basically, there are some loss of habitats of this wildlife. So the wildlife try to find new habitats, and basically there will be some interaction between the wildlife and the livestock, as well as with the human beings. And so far, probably predicted there are more than 1 million unknown viruses in this earth, on this earth, with some pandemic potential that might occur in the future. And only 1% of this more than 1 million viruses has been identified so far. Yeah. So wildlife actually a very valuable sentinels for threats for human as well as for livestock, domestic animals, including the non-communicable disease threats. And there are certain evidence or proofs that this wildlife is quite important for the possibility of disease contracting to human beings. This slide shows the importance of the three taxa, the three orders of animals, compiled by Kevin Olifel from EcoHealth Alliance. Basically, he compiled publication from several articles, maybe several decades, maybe 4 or 3 decades. And then he concluded that despite of the total viral richness in every orders of mammals, the three taxa are playing important roles in contributing to disease transmission to human beings. Those are the Chiroptera, the bats, yeah, and then the primates, and then the rodents. Yeah. So these three orders are the most important wildlife to be considered as a carrier or as a reservoir for some diseases for human and livestock. Yeah. So we are not supposed to blame these animals, yeah, but we have to be very to have a wisdom, yeah, that knowing that they are harboring some infectious disease pathogens doesn't mean that we have to destroy them, but we have to actually make our environment to live harmonious with these animals here. This model developed by Dr. Billy Carres from 2012, yeah, published it in the Lancet, and this is a normal situation that probably should be paid attention by us, every one of us, yeah, that wildlife are shedding viruses in the environment. And that would be some fluctuation of the shedding. And at some time point, it's possible that the shedding of the virus might spill over to human beings here or to livestock. Yeah. And this will cause some economic loss and probably fatalities and mortalities in the human beings or the animals. So basically, this is a model that if we just take everything as is without any prevention, without any mitigation, so this will occur all the time. We will see that the occurrence of disease in human beings or in livestock, but then after a while, we try to study further that this disease actually can be prevented when we understand that the potential viruses shed by the wildlife are identified from the beginning. So basically the second curve showing that if we do some, say, active surveillance, yeah, so we would like to see how many viruses are there in the environment shed by the wildlife. And we could identify at some time points that certain virus might be harmful to human beings in probably 1 decade or 2 decades from now. So we portray the situation now to predict the possibility of potential pandemic in the future. So if we do that, if we do the surveillance of the virus from the wildlife, then we should be able to prevent the occurrence of the spillover to livestock or to human beings. So the loss would be decreased by 60%. This is a model, of course, yeah, but in the reality, there are lots of factors that we might have to face. Yeah. This is a good example. Yeah. World Bank actually already made some value evaluation of the disease effect about the economic loss of disease emergence from time to time. Yeah. And we see here that lots of loss that's been reported, and the last one from COVID-19 is very huge, about 15 trillion US dollars. Yeah. So this is the loss that we face if we just ignore, if we just take the activities, business as usual, without any prevention, without any control nowadays. Yeah. So this one model again, this project is called PREDICT, and it was funded by the USAID, U.S. Agency for International Development, with the consortium from the U.S. consisting of the UC Davis, EcoHealth Alliance, Metabiota, Smithsonian Institution, WCS. And it consists of more than 30 countries involved with this surveillance, including Indonesia. So we basically took part in the first stage of this project from 2009 to 2014, and then continued from 2014 to 2019. Yeah. From the 1 decade project, PREDICT globally, yeah, identified about 1,100 viruses, and more than 900 are novel. And it consists from the 28 families of virus. Yeah. And this involved the samples from wildlife, from livestock, as well as from human beings. Yeah. Not just the virus discovery, but PREDICT project also have some capacity building. So basically, lots of more than 6,000 people actually been becoming like a workforce for this project. and then more than 60 laboratories in the world has been built the capacity to be able to detect the virus that previously undetected, and then more than 160,000 also operated, and more than 1,100 viruses identified. Yeah. A bit about the project in Indonesia. We also try to have some behavioral studies in human beings. And as we probably are aware, some part of Indonesia, and again, this is not just specific to North Sulawesi, but also happens in Kalimantan, in Sumatra, and somewhere else, even in Java, like Yogyakarta, Solo. Yeah, a lot of people consume the wildlife meats, the bush meat. Yeah, so we can see that the three taxa of the important wildlife that contributes disease pathogen to human being actually are consumed very regularly, especially in the northern part of Sulawesi, like the bats. These are the roasted bat, not really well cooked, yeah, not yet well cooked. But we also sometimes see primates, non-human primates. The 7 species of non-human primates in Sulawesi are actually critically endangered. And they sell this non-human primates carcass for very low price, like 50,000 rupiahs per kilo, and it will probably sell for maybe 250,000 or 300,000 maximum. So compared with the conservation status of this animal, it's very ironic to see this situation. And it's very common that they actually have this sold in the wet market in North Sulawesi. Again, I don't want to, what do you call it, discredit the situation only in North Sulawesi, because we just focus our activities in North Sulawesi, but it also happens probably somewhere else in Indonesia, and also Southeast Asian countries. Okay, so some recommendation that we also give that we have to have a massively scale up One Health surveillance across the 3 interface, the wildlife, livestock, and human beings. Yeah? So lessons learned from the PREDICT project, we should also try to have disease surveillance in more stable situation. Basically, it is important that the country ownership of this surveillance is a key. It's not going to be sustainable if we always depend upon the international agencies' funding. So basically, we have to ask attention from our government to provide support for continuous surveillance. And of course, with the variety, diversity of biodiversity, and also cultures in Indonesia, the risk is not uniform from one land to other land, or from one island to another island. So basically, we should have some kind of target surveillance to be implemented from one place to the other place. We have to identify and to strengthen our One Health workforce from field to laboratory. At the moment, this PREDICT project actually try to strengthen the capacity of the laboratory from the government of Indonesia. So 8 national laboratory for animal health or animal disease investigation has been trained for this particular laboratory protocols, as well as maybe 4 or 5 units from Ministry of Health, and several other also laboratories from the universities. These are the field activities that we conducted. So we take samples and we try to preserve. We don't want the samples to be ruined from the transportation or from the poor storage in the field. So we bring all the logistics, required logistics to field. It is not easy. Sometimes we have to also have to go on the water, yeah, not just on the road. This is almost the last slide. So I just want to point out here that common viral transmission from wildlife to human beings or to livestock are taking a certain patterns. And we would like to show you here that this reservoir horse basically are animals that are infected by certain pathogens, but they are not showing any clinical symptoms. They're not affected by the microorganisms or the pathogens. But once the same pathogens actually infect other wildlife or maybe some livestock, yeah, it will give some clinical symptoms and probably some fatalities as well. So these are the intermediate hosts that also play a role as an amplification or amplifying host. And human beings or livestock maybe is the fatal host for all the situation that it might be transmitted directly from the wildlife to human beings, but at many cases it also has to go through the intermediate host and then infect the human beings. And from this slide, we actually have several questions that we learned today about self, no self technology. The first question is how the self, no self technology could be implemented in preventing emerging infectious diseases. I think I learned from the discussion earlier that might be, we have still to think about viral RNA that might be involved in many cases, that most of the emerging infectious disease actually caused by the RNA viruses, yeah? And then we also need to consider maybe this will be able to be implemented in non-communicable disease, as well as on the antimicrobial resistance that's been discussed also earlier today. Yeah? And then we probably need to know which chain should we pay attention to, either to the reservoir or intermediate host or the fatal host. And this, of course, will need some approaches that, again, in the importance of, the sake of importance for the human host, probably we will focus on the fatal host, like human beings or livestock. Yeah. And then we also have come with the last question on the stage of disease control and prevention. Will this be comparable to something immunization that probably been introduced in the livestock or in human beings, or it might be something different, but will what do you call it? Complement, yeah, to the current situation. So again, I'm new in this technology, in this mechanism, so maybe the question is not quite representing the real situation for every Indonesian, for every person here in the room, yeah. But this is something that we can discuss further, yeah. With this, I will end my presentation. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Joko Pamungkas. Ladies and gentlemen, we already have some additional complete overview regarding the problem of health in the global stage. And from what Dr. Pamungkas has presented, we could learn something that how complicated this, what they call it, the human disease originated from animal or zoonosis. But we also noticed that he should put control on operation and also control on benefit to people to minimize the negative impact of this kind of disease. But on the last presentation, he raised challenges on how the application of no-cell technology to overcome this kind of disease who currently also we face in the global state. I think this end of the presentation for the whole day seminar for today. But for the three last speakers, I would like to give some opportunity for the audience to raise some question or comment before we move forward to general discussion that will be led by Taku. Any question or comment, please? Okay, please, on the back, gentlemen. Maybe you need with moment with your mic. Please identify yourself and to whom the question is.
Thank you. My name is Haryono Joydiharjo. My background is actually aerospace engineering, but I come here because of friend and I got to be attracted. Then I'm attracted to the topic that is very, very what is intriguing. First is One Health, and the second is no self mechanisms. Both are actually very new, but I learned actually that One Health is already, it's a universal, promoted, although it originated probably in the last decade, probably, something like that. But what I'm really very curious is about the origin or the philosophy or the basis of the technology of what you call no self mechanism. I'm sorry I came late, so I didn't really follow everything. But of course, I'm still learning, so I would greatly appreciate if that can be summarized, probably also for the benefits of other attendants here. Thank you very much.
Thank you. Pak Joko?
Haryono.
Okay. Anyone will respond this question? Oh, one by one, go ahead.
Thank you, Pak Aryono. I try to give my best answer. But we have Professor Stefano here that can provide a better answer than me and also another speaker. But for me, to best to my knowledge, that the concept, Pak Aryono, the no-self DNA, is based on the recognition of these foreign materials, the DNA, but they can act in a species specific to inhibit the growth of that type of species of bacteria or DNA. So the thing is, they work into specifically to the DNA that we fragmented. So that's the concept of the cell mechanism. So this is a novel approach in how to mitigating the disease rather than to use the antibiotics that can have the antimicrobial resistance in the tissue of the culture organism and other or plant or human. And by doing this, I think is more safe to the human because this is no reaction to the organic to the organism, and this can be applied in the future as the novel approach for One Health solution. Thank you.
Dr. Isa, you have something to add, please?
May I proceed? It's very interesting because I remember there's a professor in, I think, ITB who develops how you don't use fertilizer, but use something that originates from that plant itself to have a better growth. Like this is for agriculture, and one of my students have practiced that. Now, in that case, I like to ask further because maybe I do not completely got all the information. But that means that are you resorting actually to the DNA itself rather than to the environment or others in order to take the best advantage of that? In fact, for many positive purposes. Is that so? That you resort more on what is, so it is no self mechanism, but is it self mechanism? So if not, what are the differences? Thank you. I hope you got my questions, right? Thank you.
Okay, great.
Okay, thank you, Professor Pak Haryono. I will try my best also to answer as a scientist. So, the basis of self and non-self DNA. So basically what you're talking about or asking about is about sensing mechanism. I will give you an analogy. So if you have an organism, so the organism will cope their life from diseases, from, I don't know, stimulus, by acting in two ways. The first one is by receiving warning from themselves. That is what we call self-DNA. And receiving warning from other. Because we're talking about DNA, so we talk about self-DNA and non-self-DNA. So basically, when you have either self-DNA or non-self-DNA, in my limit of my knowledge, I do not quite understand completely how the mechanism happens, but either the both mechanism, it can triggers what we call internal system warning in the organisms in cellular level, meaning that in self-DNA, the self-DNA would like to say to your cell, there is something happen, there is your DNA fragmented or destructed or the one that is not normal according to the cell, and the cell will respond, let's stop first. Like when you have an accident in the road, you need to stop and breathe first, right? So that you will understand how to take action after. That is self-DNA. The no self-DNA, meaning that when you have a response, you see other people having an accident and you say to yourself, I need to be very careful to not having the same accident as that guy in front of the road. That is what we call the no self-DNA. But the concept itself can be used also, that no self-DNA, meaning that no self-DNA for you, but could be self-DNA for the diseases inside you. For example, you have plants. Oil palm have Ganoderma inside. So when you put the DNA of a Ganoderma, we have potentially inhibited the growth of Ganoderma inside the oil palm. This is only the analogy. So meaning that either way you choose self-DNA or no self-DNA to understand, you are facing with a sensing mechanism that has been given maybe probably a long time ago, and we have the opportunity to understand the mechanism nowadays, that you have this mechanism, you can use it to developing product, for example, if you talk about plantation or even health, developing drugs maybe that can, that based on the self or non-self DNA to induce or inhibit some mechanism in your body, or even the mechanism of other diseases generated by viruses or bacteria and etc. So I hope that I give you a more global understanding using an analogy, meaning that we have a long way to run actually with self and no self DNA. But this no self DNA technology is actually the one that we can think about, because it will inhibit the other diseases that we don't want to in our body, or we don't want to in other plant species, for example, if you, or even in fisheries. So yes, I'm quite excited with what we see today and nowadays with the no self DNA. Maybe in the future we will find another understanding about this technology. I hope that's all from me. Thank you. Thank you, Pak Aryono.
Thank you, Dr. Risha, and I hope that's already satisfied Professor Haryono for his question. Otherwise, after the meeting, you will discuss again with Dr. Risha outside. Pak Joko, you may have something to add.
Thank you, Prof. Haryono, for the question. And I think I'm going to answer about the One Health concept or approaches. So basically, if we define, if we learn about the definition of the One Health, and then a person probably will think, oh, I've been doing that for so long. I've been doing that from 3 decades ago. Yeah. So individually, the practice, the activities probably been in place, but then because of the appearance or the occurrence of H5N1 avian influenza in early 2000s, more realization, more understanding about the importance of this One Health concept or approach among the institutions. So it is not just being practiced by individual, but it should be also practiced by the organizational level and also even sector from the government. So basically, the concept is something that in order to be able to combat the human disease or livestock disease, it cannot be just practiced by one sector like Ministry of Health or Ministry of Agriculture, but we have to collaborate, we have to coordinate, communicate each other in order to be able to actually solve the problem comprehensively, not just in the human aspect, but also from the upstream. Maybe we have to deal with the ecosystem, maybe something wrong with the management of the ecosystem, something that we have to think as a holistic situation, but not individually, but more institutional organization, or maybe even government. Yeah, I think that's my explanation, Pak Haryono.
All right, thank you, Pak Joko Pamungkas. And I think we are already 30 minutes over the scheduled time, but there's no problem because this is very, very important issue that we would like to have a response from all audience here who come over to this meeting. What the next of this discussion? Yeah, we will have some discussion for general issue later, led by Dr. Taku Botama, and the target for this session is to make a networking because Ecotalis Technology Indonesia could not work alone. We need all stakeholders who may have the same interest on the application for so-called no-cell technology, for example, for agriculture and also for pharmacies. And for the scope of the global approach is for the achieving a better life for the people. And I think we have come to the last session of this afternoon before the general discussion. I would like to ask you all give a big applause for all our three standing speakers this afternoon. Thank you very much, and I'll give back the time to Ms. Anna Tsunadi for the session, the next session. Thank you.
Thank you very much, Professor Didik, Dr. Joko Pamungkas, Dr. Riza, Dr. Romi. But before we go to the next session, I guess the weather is getting a bit cold, so maybe we can all bring our cup of coffee in here. Please do. Yeah, so most welcome.